Your ears are incredible. They are your personal sound detectors, working every second you are awake. They help you hear your favorite songs, the voices of your family and friends, and the warning sound of a car horn. Hearing connects us to the world around us in so many ways. It's a sense we often take for granted, but it is super complex and totally amazing. Think about it, sound is just vibrations traveling through the air. Your ear catches these vibrations, turns them into signals your brain can understand, and suddenly, you hear a bird singing or a friend laughing. It's a beautiful and vital part of being human, connecting us to each other and keeping us safe. Inside your ear, there's a whole world of tiny, intricate parts working together. It's like a super sophisticated biological machine. The part of your ear you can see, the outer ear, is just the beginning of the journey for sound. The sound waves travel down a tunnel, the ear canal, and hit the eardrum, making it vibrate. These vibrations are then passed along by three of the tiniest bones in your entire body. They are called the hammer, the anvil, and the stirrup. These little bones act like amplifiers, making the vibrations stronger before sending them even deeper into your ear, into a place that holds the real magic of hearing. This magical place is a tiny, snail-shaped structure called the cochlea. The name cochlea comes from the Greek word for snail, and if you saw one, you'd understand why. This little spiral is filled with fluid and lined with thousands and thousands of microscopic hair cells. These aren't like the hairs on your head, they are incredibly delicate sensors. When the vibrations from the tiny bones reach the cochlea, they make the fluid inside it move in waves. These fluid waves cause the tiny hair cells to bend and sway, like seaweed moving in the ocean currents. This movement is the key to everything. When these special hair cells bend, they do something truly extraordinary. They open tiny channels that let electrically charged particles or ions rush in. This creates an electrical signal. That's right, your ear turns the mechanical energy of sound vibrations into electrical energy. These electrical signals are then sent up the auditory nerve directly to your brain. Your brain interprets these signals, and voila, you hear sound. It's an amazing chain of events. A perfect example of physics and biology working together in harmony, all happening faster than you can blink, inside your very own head. Isn't that cool? For a very long time, scientists have been trying to understand exactly how the cochlea works its magic. It's one of the biggest mysteries in the science of hearing. The main problem is that the cochlea is incredibly hard to study. It's a tiny, delicate structure, and it's buried deep inside the temporal bone, which is one of the hardest bones in the human skull. It's like trying to study a tiny, fragile watch that's locked inside a solid steel safe. You can't just open it up and poke around without breaking it. This has made it very difficult for researchers to see what's happening inside a living, working cochlea. Because of this challenge, most of what we knew about the cochlea came from studying it after it was no longer living. Scientists could look at it under a microscope, but they couldn't see it in action. It was like looking at the parts of a car engine laid out on a table instead of watching the engine run. You can learn a lot about the parts, but you can't fully understand how they all work together to make the car go. This meant that the most important part of the process, how the cochlea actively amplifies sound, remained a secret, a black box in our understanding of hearing. This black box problem was a huge roadblock for hearing research. We knew the cochlea wasn't just a passive microphone that picked up sound. Scientists suspected it was an active amplifier, meaning it actually adds energy to make quiet sounds louder so we can hear them. Think of it like a sound system that can boost the volume of a faint whisper. But how did it do this? How did those tiny hair cells work together to create this amplification? Without being able to watch the process happen live, scientists could only make educated guesses. They needed a way to peek inside the safe without breaking the watch. So the big challenge for scientists was this: how can we keep a cochlea alive and functioning outside of the body? If they could achieve that, they could finally watch the machinery of hearing in real time. They could see how the hair cells move, how the fluid flows, and how the whole system responds to different sounds. It would be like getting the keys to the safe and being able to watch the tiny gears of the watch turn. Solving this problem would be a massive leap forward, opening the door to understanding not just how hearing works, but also what goes wrong when it doesn't. And then, it happened. A team of brilliant scientists at the Rockefeller University in New York City did something that had never been done before. They successfully removed a cochlea from a mammal, in this case a guinea pig, and kept it alive and working in their laboratory. This was a monumental achievement in the world of science. For the first time ever, They had a living, breathing cochlea that they could study outside of the body. It was like taking that watch out of the steel safe, winding it up, and watching its hands tick perfectly. This was the moment researchers had been dreaming of for decades. This breakthrough was so important because it allowed the scientists to directly observe the inner workings of the cochlea. Before this, studying the cochlea was like trying to figure out a secret recipe by only smelling the final dish. Now, they were in the kitchen, watching the chef at work. They could see every step, every ingredient, and every reaction as it happened. They could finally witness the delicate dance of the tiny hair cells and the structures around them as they responded to sound vibrations. This wasn't a static image under a microscope. This was a live performance of the biology of hearing. Imagine the excitement in that lab. The team had managed to create a perfect little life support system for this incredibly sensitive organ. They provided it with all the nutrients and oxygen it needed to survive, mimicking the exact conditions it would have inside the body. By doing this, they were able to keep the cochlea functioning for hours. This gave them a precious window of time to conduct experiments, to poke and prod, very gently of course, and to record what they saw. It was a groundbreaking technique that turned a biological black box into a clear glass case. The success of this experiment, which happened on September 27, 2025, sent waves of excitement through the scientific community. It wasn't just a small step forward, it was a giant leap. It meant that many of the long-standing questions about hearing could finally be answered. How does our ear protect itself from loud noises? How exactly does it amplify soft sounds? What goes wrong in certain types of hearing loss? The ability to study a living cochlea opened up a whole new frontier for research, promising to unveil the deepest secrets of one of our most precious senses. Behind every great scientific breakthrough, there is a team of dedicated people, often led by a visionary scientist. In this story, that scientist is A. James Hudspeth. He is a professor at the Rockefeller University and a true pioneer in the field of hearing research. For decades, Dr. Hudspeth has dedicated his career to understanding the biophysics of the ear. He's like a master detective who has been chasing the clues of hearing his whole life, determined to solve the puzzle of how we perceive sound. His work has fundamentally changed what we know about the inner ear. Dr. Hudspeth wasn't always focused on the cochlea. Early in his career he studied the hair cells of bullfrogs. Why bullfrogs? Because their hair cells are much larger and easier to study than the ones found in mammals. By studying these simpler systems, he was able to make fundamental discoveries about how hair cells work. He figured out how they convert mechanical vibrations into electrical signals, a process called mechanotransduction. His early work laid the essential groundwork for understanding the much more complex mammalian ear. He was patiently building his knowledge piece by piece. Over the years, Dr. Hudspeth and his team developed incredibly precise tools and techniques to study these tiny cells. He is known for his meticulous and ingenious experiments. He essentially invented ways to listen in on the conversations of single hair cells, measuring the minuscule electrical currents they produce. His passion for the subject and his relentless pursuit of answers have inspired a whole generation of scientists. He believes that to fix something that's broken, like hearing loss, you first have to understand exactly how it's supposed to work in the first place. This latest achievement, keeping a mammalian cochlea alive, is the culmination of his life's work. It represents the ultimate goal he had been working towards, to finally see the entire system working together in harmony. Dr. Hudspeth's journey shows us how science is built over time. It's not usually one single eureka moment. It's years of hard work, curiosity, and building on previous discoveries. His dedication to solving one of science's most complex puzzles is truly inspiring, and is now paving the way for a new era in hearing research. To keep the cochlea alive outside the body, my team and I really had to become, well, biological architects. We couldn't just put the cochlea in a standard petri dish. See, this delicate organ needs a very specific environment to survive, one that perfectly mimics the conditions inside the skull. So we went ahead and designed and built a custom-made experimental chamber. You could say this chamber was a miniature life support system, a 5-star hotel for a cochlea. It was carefully engineered to provide everything the cochlea needed to feel right at home, even though it was sitting on a laboratory bench. The first and most important thing was to provide a constant supply of what we call artificial paralymph. Paralymph is the special fluid that naturally surrounds the cochlea inside the ear. My team created a recipe for this fluid that had the exact right balance of salts, sugars, and other nutrients to keep the cochlea's cells healthy and happy. This solution was gently pumped through the chamber, continuously refreshing the cochlea's environment. The temperature also had to be perfect, kept at the same temperature as a living body, which is about 37 degrees Celsius, or 98.6 degrees Fahrenheit. But that's not all. The chamber also had to allow us to see what was going on inside, so it was built to fit on the stage of a powerful microscope. Part of the chamber was made of clear glass, creating a window through which we could peer deep into the cochlea's spiraled structure. We also needed a way to create sound vibrations. To do this, we attached a tiny piston to the chamber. This piston could be controlled with extreme precision to push on the fluid, creating tiny pressure waves that were just like the sound vibrations the cochlea would experience inside the ear. The whole setup was, honestly, a marvel of engineering. It was a closed system, meaning the precious artificial fluid was circulated and filtered to keep it clean, and oxygen levels were carefully controlled. This level of detail was absolutely critical. Even a small change in temperature, fluid composition, or oxygen could cause the delicate hair cells to stop working. The creation of this special chamber was as big of a breakthrough as the experiment itself. It was the key that unlocked the door, allowing us to finally perform our incredible scientific concert on the stage of a microscope. With the cochlea alive and well in its custom-built home, the scientists were finally ready to see hearing in action. Using their powerful microscope, they could zoom in on the tiny structures inside. They focused on a specific part called the organ of Corti. This is the business district of the cochlea, where the crucial inner and outer hair cells are located. The team watched as the tiny piston created pressure waves in the fluid, and what they saw was honestly just breathtaking. They witnessed the cochlea's internal amplifier working right before their eyes. They observed how the different parts moved in a beautifully coordinated dance. As the fluid waves traveled through the cochlea, they caused a structure called the basilar membrane to move up and down. Sitting on top of this membrane are the outer hair cells. The scientists saw that these outer hair cells were not passive bystanders, they were active participants. As the basilar membrane moved, the outer hair cells began to change their shape, rapidly elongating and contracting. They were literally pushing and pulling on the membrane like tiny motors. This pushing and pulling action is the secret to the cochlea's amplifier. By actively moving, the outer hair cells add energy to the vibrations. They make the movement of the basilar membrane bigger, especially for very soft sounds. This amplified movement is then detected by the inner hair cells, which are the cells that actually send the sound signal to the brain. The scientists could see this happening. They saw how the outer hair cells' movements made the vibrations stronger, ensuring that even the faintest whispers could be picked up by the inner hair cells. For the first time, researchers were not just inferring this process, they were directly observing it. They could measure the exact movements and see how the amplification changed with different sound frequencies and volumes. It was like finally getting to see the individual musicians in an orchestra and understanding how each one contributes to the beautiful music. This direct observation confirmed decades of theories and provided a level of detail that was previously unimaginable. They were no longer guessing how the amplifier worked, they were watching it work. Now, during their observations, the scientists noticed something really interesting about how these hair cells behave. They found that the cells are always on the very edge of vibrating, even when there's no sound. They are in a state of hyper-readiness, like a race car driver with their foot hovering over the gas pedal, ready to go at a moment's notice. This special state is described by a cool mathematical concept called a Hopf bifurcation. That sounds complicated, but we can break it down. It's all about being poised for action. Imagine you are pushing a child on a swing. If you give it a tiny little push, it just moves back and forth a little and then stops. But if you push it with just the right amount of force, a little harder, it will start swinging on its own, back and forth in a steady rhythm. The point where the swing's movement changes from just stopping to swinging continuously is like a bifurcation. A bifurcation is just a fancy word for a fork in the road, where a system can go one of two ways. In this case, it can either be still, or it can oscillate. A Hopf bifurcation is a specific type of this fork in the road. It's the critical point where a system that is stable and quiet suddenly tips over into a state of stable, rhythmic oscillation. The outer hair cells in your cochlea are tuned to be right at this critical point. They are not quite oscillating on their own, but they are so close to it that even the tiniest bit of energy from a sound wave is enough to push them over the edge and make them start to vibrate powerfully. This is what makes them such incredibly sensitive amplifiers. Being poised at this Hopf bifurcation is the key to our amazing hearing. It allows the outer hair cells to use their own internal energy to massively amplify very faint sounds. It explains how you can hear a pin drop from across a quiet room. The sound of the pin dropping provides just enough of a nudge to kick the hair cells into their oscillating, amplifying mode. This is a brilliant strategy of nature. Instead of needing a huge amount of sound energy to start the process, the ear is always ready, using its own power to listen to the world. This incredible experiment isn't just cool science. It has huge implications for the future of medicine and for the millions of people around the world who experience hearing loss. By understanding exactly how a healthy cochlea works, we can now begin to understand what goes wrong when it breaks. Many types of hearing loss, especially age-related hearing loss or noise-induced hearing loss, are caused by damage to those delicate outer hair cells, the very cells that act as amplifiers. When they are damaged, the cochlea loses its ability to boost soft sounds. With this new technique, scientists can now test what happens to the cochlea when it's exposed to things that can damage it. For example, they can introduce drugs that are known to cause hearing loss, ototoxic drugs, into the chamber and watch in real time how they affect the hair cells. They can see exactly how the cells stop working. This could help us develop safer medicines in the future. They can also simulate the effects of loud noise exposure and observe the damage as it occurs on a cellular level. This helps us understand the importance of protecting our ears. Even more exciting is the possibility of testing new treatments. Scientists can now use the living cochlea as a testing ground for potential new therapies. Imagine they have a new drug that they think could protect hair cells, or even help them regenerate. They can add this drug to the chamber and see if it works. They can test if it prevents damage from loud noises or if it helps damaged hair cells recover their function. This is so much more effective than previous methods. It allows for rapid testing of new ideas that could one day lead to a cure for certain types of deafness. This research opens the door to developing brand new strategies for hearing restoration. Perhaps in the future, doctors could use drugs that retune the remaining healthy hair cells, making them more sensitive. Or maybe they could use gene therapy to encourage the growth of new hair cells. These ideas were once science fiction, but with the ability to study and experiment on a living, functioning cochlea, they are now moving into the realm of possibility. This single breakthrough has accelerated the race to find real effective treatments for hearing loss. So let's think about what this all means. The work of Dr. Hudspeth and his team at the Rockefeller University is more than just a fascinating discovery. It is a perfect example of the power of human curiosity. It shows how, by asking a fundamental question, how do we hear, and pursuing it with passion and creativity for decades, we can uncover the deepest secrets of the natural world. This breakthrough was not the result of a single lucky moment. It was built upon years of careful, patient, and brilliant scientific work. It's a testament to the process of science itself. This achievement will change the way we study hearing forever. Textbooks will be updated. Young scientists will be inspired to enter the field, armed with new tools and new possibilities. The ability to keep a complex sensory organ alive outside the body is a technique that could be applied to other areas of research as well. Perhaps one day, scientists will be able to do the same with parts of the eye or other delicate organs, opening up even more avenues for discovery. This is how science moves forward, with one breakthrough paving the way for many others. Most importantly, this research gives hope to people everywhere. Hearing loss can be isolating. It can disconnect people from their communities, their loved ones, and the world of sound that adds so much richness to our lives. For a long time, the main solutions have been hearing aids or cochlear implants, which are amazing technologies, but don't repair the underlying biological problem. This new research shines a bright light on a future where we might be able to prevent hearing loss or even restore natural hearing by fixing the biological machinery itself. The journey from a basic question about a bullfrog's ear to keeping a mammalian cochlea alive on a lab bench is a powerful story. It reminds us that investing in fundamental science, science driven by pure curiosity, can lead to incredible practical benefits for all of humanity. The world is full of amazing and complex wonders, many of them right inside our own bodies. By continuing to explore, to question and to experiment, we can better understand our world, and in doing so we can make it a better and maybe even a less quiet place for everyone.